So it's a real pleasure to be here, and I'd like to start with asking if you ever had problems finding your car in a parking lot like that. Okay, I see a few hands going up. If I ask this question to my students, they don't know what I'm talking about. So that's really one of the main problems that faces us as we get older. We have problems encoding these types of memories. We call them spatial memories, where we typically use a sign, like a signpost or a lamppost or a storefront to make a map in our brain that we can then instantly retrieve when we need it. But that works only when we're young, and as we get older, it becomes harder and harder to form these maps and to retrieve them. Now, it's interesting that animals actually have the same problem. Your dogs, when they get old, they have the same problem. And mice have the same problem. So we can actually use them to study some of these problems that are associated with aging. So what happens then with this brain, with this supercomputer that functions so well when we're young, but as we get older, it starts to lose its capacity to form memories, to retrieve memories, and it starts to get highly susceptible to diseases such as Alzheimer's disease and others. We know, of course, that the connections between the main brain cell, the neurons, start to degenerate, they start to disappear as part of a normal aging process. You even lose some of these neurons. And the brain really becomes susceptible to degenerative diseases. It actually also shrinks. In a 90-year-old person, the brain has lost about 10% of its volume. Now, the population is rapidly aging globally, actually, not just in developing countries, but across the globe. The fastest growing segment of the population are people older than 60 or older than 65. So this really is something that we have to start to understand and tackle. We have to be able to understand why brains start to deteriorate when we get older, because not just our countries, but developing countries in particular will not be able to bear the economic burden that comes with the care for people who can no longer take care of themselves. In Alzheimer's disease, you often need one person to take care of a patient. And so it's a tremendous economic burden and, of course, social burden and emotional burden for societies that we need to really address. So as I already said, the brain becomes susceptible to diseases such as Alzheimer's, and it's really illustrated in that slide very nicely that as we get older, the rate of Alzheimer's disease dramatically increases. As you can see in this graph here, around 65 to 96 years of age, there's only about 3 to 5% of the population have Alzheimer's disease. But as you get older, age 80 or 90, there's up to 30 or 40% of people have the pathological and clinical symptoms of Alzheimer's disease. The Alzheimer's Association estimates that if you could delay aging by just 5 years, you would get rid of almost half the cases with this disease. And that is really because age is the key risk factor for this disease, and we'll come back to that again and again. So why don't we have treatments for Alzheimer's disease? You may have seen that another very prominent trial was stopped by Biogen today because it just occurred, the analysis just showed this will not show any benefits, and so they stopped this trial. So hundreds of millions of dollars, billions of dollars have been spent already to try to find treatments for Alzheimer's disease, and yet we don't have anything that works. So I think one of the main problems is that the brain is of course isolated in our head. It's not something that if you have cancer, then frequently you can take a biopsy, you can take the tissue and you can study that tissue. You can even in the same patient, you can study the tissue repeatedly over time. But the brain is very difficult to get access to. So most of what we know about the brain is either through psychiatric or psychological analysis to interactions with people, through imaging where we know structural changes, or from analysis of brains of people who have died. So most of what we have learned is really from brains of dead people who had a disease. But that's often, in the case of Alzheimer's disease, we think this disease takes 10 to 15 years until you see clinical symptoms. And so the whole disease course may be 30 years. And when you get the brain, when a person dies, you look at what happened over 30 years. But that's, of course, too late. So we have a limited understanding of what really goes on in these brains at the molecular level, and just in regular brains of normal, healthy people, because we can't take biopsies from the brain. The other main aspect, I think, is there has been too much focus on rare genetic forms of Alzheimer's disease, in my view. We learned a tremendous amount from those. But most people don't have these rare genetic forms. They have what we call sporadic forms, and I'll elaborate on this a little bit more. There's also an increasing focus on very rare variants that increase your risk very minimally, but don't really have a big impact on the disease. And that, I want to stress that point here. So what we look here is a graph where you list here on this axis how many people have a gene. So it is from very rare to very common. And here on the y-axis, you list the relative risk for the disease, from very low risk to medium risk to high risk to you will definitely get the disease. We call these autosomal dominant genetic diseases. So there's a few mutations that occur in very rare cases that when you have this mutation, you will get the disease, 100%, unless you die, of course, before you get the disease. And then there's a few that are sort of in between, but there's a lot of genes that have recently been discovered that are very common. All of us have one or the other variant. And these variants, they increase the risk to get the disease often only by a few percentages, 1%, 5%, maybe 10%. But they don't really have a big impact. What you eat or how much you walk or how much you read has probably a bigger impact than most of these genes. So what is the biggest risk factor for Alzheimer's disease? What do you think? Aging. So aging is very common. We all get it. Biggest risk factor, it has a very high risk, as I showed you in that statistic. At age 80, 85 or so, roughly 25 to 30% of the population have Alzheimer's disease. So very high risk age. And as we get older, there will be more and more people. So what is aging? What happens actually with aging? You can look at this from a scientific perspective. There's a lot of scientists who study the molecular biology of aging. You might have heard about some of these pathways that change as we get older. This is a great review for those who want to read up on this. And it lists the key hallmarks of aging. You may have heard about telomerase. This is an enzyme that basically shortens the DNA every time the cell makes a copy. So if you make too many cell divisions, if you make the cell divides and divides, it comes to an end and it can't make new cells anymore. So that's part of aging. DNA mutations, as we get older, the DNA starts to accumulate mutations. Or some of the proteins become unstable. The mitochondria, the engines in our cells, don't work as well. Basically, everything goes down the drain as we get older. So, yet we can actually put our finger on it. There's molecular changes that we can measure. So we call these individual hallmarks of aging, and then there's two that we call integrative hallmarks. They're the result of everything that happens here. One of them is the stem cells in our tissues exhaust. They basically become inactive or disappear. And the other one that I will focus on is called changes in intercellular communication. What does that mean? I communicate with you here by using words. The cells in our body communicate with each other, frequently with proteins. And we call this intercellular communication. And that changes with age. And it changes because of all these other things that happen. So it's an integrative change. If we look at the change in this communication, maybe we can learn something about the aging process, and I'll come back to that later. Now, this is the molecular view of aging. If you just look historically about age, what do you think is the average lifespan of humans in a natural environment? Not in this sophisticated environment, but if you looked historically, 5,000 years ago, how old did humans live? Or even 2,000 years ago? 35, 40 years, 30 to 40 years. So that's the lifespan of humans. This is shown here in this very instructive graph. So this is time 0, and this is today. And basically, until about 100 years ago, people lived up to their 30s or 40s. That was the average lifespan. There were always individuals. We have historical documentation of people who lived for a very long time. They lived up to 70, 80, or even 100 years, potentially. And there's documentation for such individuals, but they were extremely rare. So why do you think that change happened? What is the most important reason why we have this tremendous jump here? And you can actually see it here. From 1900 to 2000, we go from roughly age 45 to 50 up to 75 or 80 years of life expectancy. The main reason is really the hygiene and immune function. The vaccination that allowed us to basically overcome a lot of the common diseases that killed people very early on. And then hygiene to basically protect ourselves against these bugs, bacteria, viruses that would usually kill people very early on. So that's why we escaped this natural selection, if you will, that killed most people at this early age. So the additional 50 or 60 years of life that we enjoy now are what we call no longer under evolutionary pressure. So evolution has not selected for a long life. Evolution selects for reproduction. And it was enough to have 30 or 40 years of life to reproduce. And we'll get back to that in a few slides. So let's look again at this change in age span. Most people in the past, when you look here, they probably died relatively healthy. I mean, they were very healthy. They got a deadly disease and they died in a short period of time, typically. Whereas now, our picture is very different. We have, on average, maybe 50 to 60 years of a healthy life where we feel no pain when we get up in the morning. And then increasingly, as we get older, there's all kinds of aches that start to accumulate. Blood pressure, heart disease, diabetes, brain doesn't function as well, and so forth. Arthritis. So roughly 25 years of your life, if you live up to 80, are a combination of different diseases, and they start to accumulate so that people in their 80s often take multiple drugs against all kinds of age-related diseases. Pretty depressing, right? Now, there is sort of a natural experiment in people we call centenarians, or people who live to very old age, could be 90 or 95. Many of them, they actually never get sick, or they have none of these age-related diseases. Many of them just one day they say, I feel tired, and they want to say goodbye to their family, and they pass away. There's many stories like that of centenarians. They're basically healthy. I mean, they don't run around like a spring chicken, but they don't have these age-related diseases. They don't take all these medications. So that means it's possible for the human body to live healthy for 100 years. And many of these people, when they get sick, they have a very short period of time of sickness and then they die. There is a joke that once they see a doctor, they die. So what our field in aging research is really starting to try to do is to mimic these centenarians. So that if we live to 80 years of age, that we could go from this planet like them, that one day you're just tired and you fall asleep, but you don't have 5 different diseases to get there. So nobody wants, of course, extend lifespan and at the same time make this period of suffering longer, that you would have 50 years of being sick. It's of course possible that if we did achieve this, that people would live longer as well. So there would come an increase in lifespan. But I think anybody who's serious about doing research in this space would never expect that all of a sudden you would live 50 years longer or 100 years longer. That's just an illusion. But it's definitely possible to start to understand what happens in these centenarians. Why do they live so long and are healthy? And can we start mimicking that? I want to give you a few more vignettes of why aging is actually not set in stone, but we say it's malleable. It's a process that can be influenced. These are very sad statistics from the US. They were published in the Washington Post last year. And this basically shows life expectancy at birth, depending on which county and which state you're born. So if you're born in Hawaii, you have the highest life expectancy overall. But within Hawaii, depending on which county, your life expectancy may be 12 years shorter in the lowest neighborhood, or it may be 10 years longer. So it goes from 70 to 92. Now, sometimes there are differences, there are racial differences that account for that, but not always. It's oftentimes other factors. It's socioeconomic factors, not racial factors, that are responsible for that. And you see this in every state. And not all of these states are racially very different. Some of them are, of course. But look at New York. You can have the lowest life expectancy is in their 50s, and the highest is in the 95s. I actually work with Nir Barzilai in New York. He studies centenarians, which just have their families just have very high life expectancies. And they live in a very tight society with people that sort of provide a support network. Now, we don't know exactly why these differences occur, but I just want you to appreciate that how long you live is a very flexible, very malleable process. So when you're born, only about 25% are set in genetic code how long you will live, and the rest seems to be environment. Now, as you get older, your genetic composition will determine how you respond to the environment, but still, it means a lot of what influences how old you are is determined by the environment you live in. How you live your life, but then also environmental factors. Now, my third point is one of these most amazing experiments that you may have heard of. So what Shinya Yamanaka, who used to be a postdoc actually at the Gladstone Institute before he went back to Japan, what he discovered is that you can take an old cell you can express 4 different genes that are now named after him, the Yamanaka factors, and you can turn this 90-year-old cell into a cell that has age 0. With this cell, you can make a new organism that is exactly like one that was conceived from sperm and egg. We call this reprogramming. You may have heard of induced pluripotent stem cells. This is a process where you turn old cells, skin fibroblasts, some of you may even have donated at the hospital in research studies, skin fibroblasts. What we can do with these, we can turn them back into young cells that have age 0. What does that mean? That means that age is malleable. You can take an old cell and you can make it completely young again. And my last point of something that has actually eternal life almost is what we call the germ line. So the germ line is the cells that keep reproducing and maintain a species in perpetuity. I don't want to say maybe forever, but in perpetuity. Maybe the same thing. So if you have a child that is conceived, you get a fertilized egg. That fertilized egg has age 0. Even if the parents are 30, 40, or even 50 years old, the child will be age 0, right, when it's conceived. When it's born, we call it age 0. And it grows up, 2nd generation, 3rd generation, 4th generation, 100 generations. This here is always age 0. So that means age does not necessarily accumulate. If you think about that, age does not necessarily accumulate. It's a malleable process. If you think about this in another way, our genetic code has a written information that makes an organism such as ourselves. But it's all based on building blocks. And if you have the same genetic code, you can always make a new protein, a new Lego block that is age 0 at any time. It's not necessary that age accumulates. You can erase it. And that's sort of, I go back from our discovery to try to explain it up front, but it may explain what I show you in a few minutes. Why can we slow down or reverse aspects of aging? Because aging is really something that is plastic, is malleable. So I just said that. And based on some of these discoveries, people have appreciated that aging may be a druggable target. That for the first time, in like 5 or 10 years ago, there were companies started that said, we want to target aging. Some say eradicate aging, but that's silly. I think what we want to do, as I showed you before, is we want to slow down the aging process. We want to enable you to live a healthy life until you die. Increase health span. And these are just, you can't read this, but it's just a list from companies that focus specifically on the process of aging and try to intervene in that to specifically target then diseases of aging. Now, there are some companies, well, as I said, that want to target aging per se and promise you life forever and things like that, but I think this is mostly silly and is not based on scientific evidence, but there is serious science that shows that we can start to understand the aging process, slow it down, or even reverse it. So what if there was this magical fountain of youth? This is a painting from Lucas Cranach the Elder, this in the Gemälde Gallery in Berlin, and it shows these old and frail people here entering this fountain. They emerge on the other side and they're young and rejuvenated. So wouldn't that be wonderful if that existed? Where is that fountain? Yeah, there's a lot of people actually in every culture, every tradition, there are stories like that. Sometimes it's a fountain. Ponce de Leon was looking for the fountain of youth, right? And he sailed in much of his life, apparently in the Caribbean, to try to find it. In Florida, there is actually, I think it's near St. Augustine, there is a fountain of youth where you have to pay entrance and you can drink the water, but I haven't heard that it had helped anybody so far. Alexander the Great, for example, or in China, Xu Fu, who tried to find the fountain of youth, and then the story has it, he actually landed in Japan and stayed there because he couldn't bring the fountain of youth back to the emperor. So what I want to tell you that is, in mice, we may have found something like a fountain of youth. And that's with this model called Parabiosis. So Parabiosis means living by sight. It is a surgical technique that was first done in France about 130 years ago to study transplant rejection. And so they wanted to find out, can you put a tissue from one animal to another, and it helped actually to establish the basis of organ transplantation that we use today. It's a little bit creepy, I have to admit, because what you do essentially is we suture two mice together at the flank, and because this is a wound, the blood vessels grow into that wound. They fuse with each other, similar to a placenta, or a little bit different. And now you have an exchange of blood from young animals to old animals and vice versa. It's somewhat similar to what you might see in Siamese twins, where the blood circulation of two children is fused. Now, what you have then is basically blood that connects now all these different organs in the two animals. And Tom Rando, who's a faculty colleague, professor also at Stanford of mine, he has used this model in recent times to study the aging of stem cells, specifically of stem cells in the muscle. So when you get older and you have a hard workout, your muscles actually do not produce new muscle tissue. But when you're young, that works very well. Bodybuilding. When you work really hard on your muscles, they start growing. That's because you activate the muscle stem cell, and the muscle stem cell then makes new muscle fibers. But for some reason, these stem cells become inactive in old organisms, in old mice, in old people. And Tom had the very simple question, is this because something inside the stem cell is changing with age, or something outside? And the way he could test that is by connecting an old mouse with dysfunctional stem cells to a young mouse. And what he saw was really most remarkable. He saw that the old stem cells would just regenerate muscle tissue like a young one. If you looked at the tissue, you couldn't see a difference between the young and the old. It was really magical. And at the same time, he also saw effects on the liver, and he had some indication that the brain would also benefit. And that's how we started to collaborate with each other, and could then show, and this is also work from many other labs now, that muscle, pancreas, liver, heart, brain, bone, kidney, and the growing number of tissues seem to benefit from that, where the tissue of the old animal starts to look younger, functionally, but also at the cellular and at the molecular level. So how does that work? The blood is, of course, the tissue that connects all the different organs in our body. So what if the blood actually carries important information about aging? I'll show you in a minute what I mean with that. So can we study how aging occurs in our body by simply looking at the blood and ask, how does the blood age? Can we learn something about the aging process from looking at the blood? I'll show you just one example of how we do this. This is a relatively small study with 170 healthy people who gave us a blood sample. And we measured 1,300 proteins. 1,300 proteins, most of them involved in this communication that I mentioned before, intercellular communication. So this would be hormones, like insulin is a communication factor between cells. You may have heard about interferons or other cytokines, interleukins, factors like that, TNF-alpha. So these are factors that cells use to communicate with each other. So we measure many of these and see, does their level change when you get older? The idea is that we discover a signature of aging, and by looking at which factors make up the signature, we learn something about the aging process. And so again, as I mentioned, we look at this aspect here of aging, this communication between the cells. And this is how it looks. So we can represent these changes in beautiful colors. So here are these 170 individuals on the x-axis from 20 to over 100. And here are the 1,300 proteins. And then in blue color, we show proteins at low level, and if they get warmer and go up, they become yellow. So what this graph, or what this image essentially shows is that there are a lot of proteins that are present at low levels in young people, and they increase as you get older, and they're much higher when you're old. Many of these proteins are inflammatory factors or injury factors. So these are factors that are probably not very good for your body. Other factors are high when you're young, and they decrease as you get older. Many of these are factors that we know are necessary to maintain tissues, growth factors, for example. So there are dramatic changes occurring simply in the blood. And as you'll see in a moment, if you give me a blood sample, I can tell you pretty much how old you are based on the composition of these factors. We have now up to 5,000 proteins we measure, and in several thousand individuals, and we see the same thing happening. So what we typically see that to model or to predict age, you actually need only a few proteins, and that's relatively trivial. If something correlates strongly with aging, you can predict the age of the organism. And roughly a quarter or a third of the proteins change from the youngest people to the oldest people, again suggesting that there are dramatic changes occurring just in our blood. And so what does that mean, these factors change in the blood? Remember I showed you this circle with all the different tissues, and in the middle is the blood? So the blood gets these proteins from all the different tissues. It collects information and distributes that information to different tissues. But it means also that as you get older, that communication is very different. So your body has to almost relearn on a continuing basis what to do with that information. It may also tell us how our body changes with age, and we may be able to learn something, how it becomes susceptible to diseases of aging. What you can do then is you can try to predict age. So here, this is what we do. We take the top factors that best correlate with age, and we calculate for each individual what their relative or biological age would be. So here then is the actual age of a person, and here is the predicted age. So if we would predict perfectly, everybody would be on this 45-degree line here, right? Then we would perfectly predict your actual age. But we know that's not the case, because we know there are people who seem to age slower, and others seem to age faster. So is that what we're predicting here? So this individual is 70 years of age, but is predicted to be only 45. So is this one of these people who doesn't seem to age as fast and look much younger their age? Show-offs. And is it maybe a person who lives to be a centenarian and will have a very long life without getting any of these age-related changes? On the other hand, this individual is not even 40, but predicted to be 65. Is this a person who has maybe an underlying aging process or disease process, age-related disease process already underway? And if we could detect that, could we start recruiting people into treatments before they actually get age-related diseases? Now, this is still experimental, but we can use this information and test it now, for example, in animal models, in mice or in fish. We work with fish that live only about 6 months, so that allows us to rapidly test some of these factors. But it gives us new ideas of why there are differences in aging, in biological aging in individuals, and what these factors might be and what they might be doing. So, but still, to be honest, this is all correlation still. It's like these beautiful stories you always read in the New York Times, that if you eat more of this, you get more of that. And you never know, is it cause or is it effect? Or is it a correlation, or is it something that is really causing biology of aging? So we want to know, are these changes that we observe with age actually modulating the age of the organism? And that's where we come back to this model, parabiosis, because that allows us to take the young blood, put it in an old organism, or vice versa. So we can test whether the blood of a young person has a beneficial effect on an old mouse, for example. And I'll show you how that works. What we discovered is we actually don't need to suture the animals together. We can collect blood from many young mice. We get rid of the cells because we found for the effects on the brain, we only need the soluble factors. We call this plasma. The soluble fraction of blood is called plasma. We repeatedly inject this then into old mice, and what we see is that there are more stem cell activity, so the stem cells get rejuvenated. There is more synaptic activity. The neurons in the brain start to function better. They communicate more. There's more electrical activity, and that's a correlate of cognitive function. And there's an attenuation of brain inflammation. So as I mentioned to you, many of the factors that go up with age have to do with inflammation. And our brains actually are increasingly inflamed as we get older. And when you treat these old mice with young blood, you can reduce brain inflammation. On the other hand, the young companion in the parabiosis model actually suffers from this exposure. And we can show the same thing if we collect blood from a lot of old mice, inject it into young mice, then we can reduce neurogenesis, we can reduce the stem cell activity, you can reduce the communication between the neurons, we get more inflammation, and we can also impair their memory function. We isolated, based on these findings and measuring the proteins that change with age and then are reversed with young plasma, we identified a couple of factors, or 3 factors here actually. One of them is now targeted in clinical trials by the company we started and had promising phase 2 results in age-related macular degeneration. So we think you can actually discover factors that are relevant for aging and that may even have therapeutic effects. We have to go one more step, a large phase 3 blinded study to really prove that this works. But it looks very promising. So I just want to go back to this model here and show you how these old mice actually benefit from getting young blood. And what we did here is we used sort of a parking lot test for mice. So you remember when you park your car, you have these different spots where it is, and you have to remember where you parked it, right? So the mice here, they have to learn where the escape hole is. So let me explain to you how this works. It's a table that is about 3 feet wide, and it's about this much above the ground. And there's bright lights shining on it. So if you put a mouse there, it wants to escape. It really doesn't like that environment. And we have a lot of holes in this table, but there is one hole where we mount a tube underneath. And we teach the mouse that it can escape into that tube, and it feels more comfortable in there. It uses, like we use the storefront or the lamppost to make a spatial map, the mouse uses here a checkerboard, a triangle, a rectangle, and forms these spatial maps. And when the mice are young, they learn this pretty fast. And we actually rotate the location of this every day. We do 4 trials, 4 days. And so the mice have to be pretty smart to figure out every day, oh, it changed again, different location, where is it today, and remember that. So let me show you an old mouse. You can see it here. That was treated just with a salt solution as a control. We taught this mouse 4 days, 4 trials per day, that the escape hole is here, but it can't remember. It just doesn't find it. It doesn't know where it is. It's too hard for this mouse. Now the next mouse I show you is the same age, litter mates, but it was infused every 3 days with a small amount of actually young human blood or plasma, because this allows us to find the human factors. It would be the same if we used young mouse blood. Let's look how this mouse performs. So here's the mouse again. It almost shows a different behavior, a different strategy. It's the best mouse we ever had. So you see a clear effect of injecting this young plasma, this small amount of plasma that we inject over 3 weeks, every 3 days. And if you want to see the statistics, so we had here a relatively small group of mice, 7 and 8 mice per group. Again, 4 days, 4 trials per day, and you see these old mice really don't learn the task. But if you treat the same old mice with young plasma, either from humans or from mice, they start to show trends in the 3rd day, and then on the 4th day, they become highly reliable. Every mouse actually can master that test on the 4th day. Now, I mentioned to you that because this was so translational, and plasma is of course given to people all the time. I went to conferences and talked about this. I'm myself not a clinician, but my clinician colleagues who study Alzheimer's disease said, why don't you try this in humans? Humans get plasma all the time in the hospital, and it should be straightforward. So that's what we did. We started a company and got funding from actually another big firm that makes plasma products, that makes albumin or immunoglobulin fractions out of collected blood from donors. And what we did is we did a trial that was run here at Stanford by Sharon Shah. We recruited 18 patients with mild to moderate Alzheimer's disease, and we gave them 4 units. So once a week we gave them a pint of plasma from young donors from the blood center here at Stanford. You had to match by blood group for this experiment here. And this is a so-called phase 1 study, a safety study, so it's very small and you can't really make any predictions of future results based on it. We still looked at memory and some activities, and there's some interesting trends. You could read the paper if you want, but again, we can't really make any conclusions at this point. Most importantly, it was safe. We didn't have any unexpected adverse effects, and that's why we moved then into phase 2 trials. And the company we started, Alkaest, has now phase 2 trials in mild to moderate Alzheimer's disease, in severe Alzheimer's disease, which has almost been completely abandoned by pharma industry. But we believe that even in a progressed Alzheimer patient, that there might be something that can be recovered based on the molecular changes that we see, the profound molecular changes we see with young plasma factors. And so this is now in clinical trials, also in Parkinson's disease, I forgot to say, based again on preclinical data in mice. So for a lot of people, and I can totally understand that this sounds almost too good to be true, right? How could it be so simple that you can treat potentially a disease with something trivial as just young plasma, young blood? So that triggered really a lot of questions, and we have to, as a community, work on this to really find why does it work, what are the molecular bases. But as I led up to the discussion today, to my talk, I showed you why I believe age is actually not something that is set in stone. It's not a train that just goes down a track and cannot be stopped or cannot be slowed down. In fact, that train can potentially even go into reverse gear. And so what we want to understand is what are the factors. I showed you a few factors that we find have a negative effect that we can block, that we can neutralize. We want to know where do these factors come from. As I mentioned before, the blood collects information from all the different tissues in our body. So if we see these factors changes, where do they come from? If we know where they come from, maybe we can target them already in that tissue, in a specific tissue. Which cell types does the blood rejuvenate? Only the stem cells, or maybe only immune cells, or every cell type? We have increasing information now at the molecular level that many different cell types benefit from that. The cells that form your blood vessels, for example, the cells that protect our brain, the immune cells in the brain benefit from this, as well as the stem cells. And then that brought us to a very fascinating question. So we always look at a person and we say, oh, you still look young, right? And we look at the skin. But does the skin actually reflect the age of your heart or the age of your liver or of another organ? We don't know that. It's surprising that we don't know. We should know this, right? So we're doing our studies in mice where we look over the lifespan of a mouse. We collect tissues at different ages and look whether they all change in synchrony or whether there are tissues that change faster or slower, and whether there are actually cells in different tissues that age faster and slower. And that can get you to this very intriguing possibility that aging may even spread through the body. What if there is a cell in your kidney that starts to age faster, that starts to age at age 40, and it produces one of these toxic factors that I showed you into the blood, and that then triggers aging in another tissue? Very intriguing questions that are fascinating us and that we want to answer. So with that, I would like to acknowledge my lab here, collaborators, and then funding sources, and thank you all for listening. And I'll take questions.
Thank you, Tony. Yeah, that's questions right here.
Does your work suggest that it's most likely beneficial to take anti-inflammatories on a regular basis?
So the question about anti-inflammatories comes up often. There are studies that show some benefits, but I think there are probably too many risks with most of the anti-inflammatories we have, because they have multiple different effects. Some of them are bad for the stomach or even for other processes. So I would not recommend it. But I'm not a doctor. But I think your doctor doesn't recommend it either. I think you only take them if you have to. I would not take. I come from Europe. We take much fewer drugs on average than people in this country. So I would only take a drug if you have to.
I would like to, you mentioned that there are few age-related diseases like macular degeneration and Alzheimer's, I think. So the treatments that you're talking about, do they indicate that it's a wholesale approach, or that it's specific to one kind of age-related diseases? The other question that I have, there is a big hullabaloo about human growth hormone. Is there any truth about it?
Can you repeat the second question?
There is a lot of advertisement about human growth hormones in pills. Mm-hmm. Is there any truth about do they really help?
Yeah, so maybe first the question about whether this is sort of a general effect that would target everything. I don't think so. I think what we will find is that some factors will be beneficial for one tissue, for one disease, and others will be better for another disease. There will be good factors, there will be bad factors, and I think it will be complicated. That's how nature usually is. The beauty of using plasma is that it contains thousands of different factors, right? I showed you 1,000, but there's probably 100,000 different factors in there. So it's sort of nature's elixir. And maybe that's why it actually is so powerful, because it triggers many, many different pathways at the same time. Different ones in different tissues, different ones in different cell types. And that's why it might be beneficial in a lot of different indications. And if you took just one factor, like growth hormone, it would actually only work on one specific aspect of aging or of a disease. And it would not be working. And there's indeed a growing recognition in age-related diseases, and Alzheimer's disease in particular, that it may not be sufficient to use one drug, one target alone. There's increasing discussion about a multi-pharmacy approach to these diseases. That's, of course, a nightmare, because how are you going to regulate that at the FDA if you would have to test 3 or 4 different drugs, especially if they're new? You could never, not with the current regulations, we couldn't do that. Whereas plasma is a natural product. So what we did actually in the company is we took this whole plasma, and if I may, this question may actually come up. There's a fractionation process. So there's an industry that collects plasma from thousands, tens of thousands of donors on a daily basis. And they only collect the liquid, so they don't take the cells. It's called plasmapheresis. So your blood goes in a machine, it gets spun down, the cells go back, and they take the liquid fraction, the plasma. Then they pool it and they fractionate it. And why do they do that? Because they want to extract, for example, antibodies, we call them immunoglobulins, and these are treatments for people with immune deficiencies, with autoimmune diseases, or they're even using this from people who are protected against Ebola. They take these immunoglobulin fractions and give it to people who got infected. They isolate, for example, factor 8 that is deficient in hemophiliacs from these donors, or they make albumin, which when you go to the hospital and you need a lot of, you lose a lot of blood, you may actually just get albumin and not whole blood. So these fractions are all being produced, and Alka has tested all of them in ones have the effect. And we isolated one fraction that is as good or even better than the whole. And that one is now in phase 2 trial. So we don't use the whole plasma anymore, but there's still a few hundred proteins in there. And I think that's as I said before, I think that's the advantage, that you don't have just one factor, but you have many different factors that you're using here. Growth hormone, I think, again, the dangers of using something chronically as powerful as a growth factor is probably not a good idea. But there's studies, animal studies that show that there are benefits from growth hormone. But do you want to translate that into humans who are not actually sick? And if the biology is not understood enough, I would probably not do it.
So there's lots of questions here. I want to be mindful of the order. So we have one here, then we'll head over here. There are 2 questions here, and then over there.
You may have answered this already, but in the experiment using the plasma from the young mice to the old mice, was that using whole plasma or was that fractionated plasma? And so how was it fractionated?
So all what I showed you here was with whole plasma. In my lab, we actually didn't fractionate it. We just take the approach of trying to identify proteins that change and then pick individual ones or block them. So we haven't tested fractions in my lab. That's all been done in the company. But you're probably getting to, is there a fraction that is particularly detrimental? That's probably possible also.
Over here, to your left. To your left.
Oh.
Hey, so I guess I had a question. I guess this is for the mice studies, but I'm not sure if you see something similar in the humans. But how long after you were infusing, in the case of the mice, the young plasma, how long afterwards, when you stopped infusing, did the beneficial cognitive or other effects last, or did it rapidly return to being old again?
Yeah, that's a great question. So how long does the effect last? In mice, what we and also the company has seen is that effects on the stem cells, for example, last for several months, up to 3 months or longer. If we look at other type of changes in individual cells, they seem to also last for an extended period of time. But I think they do start to disappear. So in the clinical trials that are ongoing now, what they decided to do is to treat people once a day for 5 days, wait 3 months, test them, and then give them another 5 infusions daily, and then again 3 months and test them again. So that's currently the regimen. And that's sort of based on the results in the mice. But it seems, it's clear that it does not just disappear once you stop treating. But at the same time, we don't see you treat with young plasma and the effects will last forever. That's also not going to happen. So I think it's a very much a pharmacological effect. We can actually dose it also. If you give more, you have more of an effect. So I think in that sense, yeah.
Yeah, a question here.
How have you thought about how you're going to scale this up if your clinical trials turn out well? And what is the business model going to look like? Are you going to buy young blood from younger people? Is insurance going to pay for it, etc.?
Yeah, I'm probably the wrong person to ask that. but these are great questions, and we certainly have thought about it. So as I painted the picture initially, it's daunting how many people already have Alzheimer's disease. There's about 5 million in this country, and how many will have it, as the populations get older globally. So I think if you found an effect with this type of treatment in a phase 3 study, and the effect was really solid, I think you could recruit a lot of people to donate blood for their families. They would do it for free. But it of course needs to be processed, and there is a business model behind this. But I don't think, I think there will be shortages. I don't think if this worked for multiple different diseases and for multiple different organs, it would not be a sustainable model. So we will have to figure out what are the factors, can we produce them synthetically, and maybe make sort of a cocktail of 5 or 10 factors that are most important, and block some of the worst factors. But I think it's a start. I'm particularly excited about the possibility of seeing an effect to really convince the community, the scientific community, that there is biology behind this. Because I think if people realize this really has an effect on an age-related disease, then hundreds or thousands of people will come in and start investigating how this works. Pharma will start putting money into it. How does this work? And then hopefully we'll figure out new types of treatments. But it's definitely a concern.
There's a question to your right here. Yeah.
When you say young blood, what are the age range for young people?
Yeah, that's a great question. So what does it mean young? Forever young, right? It's how you feel. So it's hard to say, but we see that when we look at the proteins, how they change with age, probably below 30 or 40 is still a good age. And in fact, from these companies that collect blood donations from tens of thousands of donors, their average age is 35, and that's what is now in the clinical trials. So I would probably not recommend transfusing plasma repeatedly from a 70-year-old, but there may be 70 years old who have very good plasma, but probably that cutoff of 40 or so is probably a good one.
Yeah, we're right here.
Have you considered the effect of senescent cells on aging, and how does that relate to the work that you're doing?
Yeah, so I brought this up here in the last point, can aging spread through the body? This relates to this question from this gentleman. So there is scientific findings that came out that show that there are certain cells that seem to change into sort of an aging stage. We call it a senescent state. They don't divide anymore, and they don't really seem to do much, but just producing some of these pro-aging factors that I showed you that go up with aging. And these cells are called senescent cells. In animal studies, if you delete these senescent cells with a genetic trick or even with some drugs, the mice actually recover in function, and they seem to be rejuvenated as well, suggesting that these senescent cells may be detrimental. And based on these findings, there's actually companies have been started to develop drugs that would specifically kill these cells. So I think it relates to what we're finding. And I think people agree it's not going to be all about senescent cells. There's going to be all kinds of different cells that are detrimental, but the senescent are probably a good target as well.
There was a question in the front here, right?
I've read a number of articles recently about how socialization, reading, brain exercises, physical exercise, vitamins, diet, etc., have been shown in small studies to reverse Alzheimer's. How might this relate to the factors in the blood?
Yeah, I think I would be cautious because they're very small studies, and they're not really. I think they're not really accepted by the neurologists, the psychiatrists in the field who study Alzheimer's disease. But what is really interesting, I mean, the concept of it is very intriguing, and I think these interventions they're definitely very beneficial, and they're probably most beneficial in the preventive state. So if you look at where do people live to 100 years, what is common to, they're called blue zones. Where do people live to very long lives on the globe? There are some in Sardinia, in Japan, in Cache County, in I think in LA there is an area where people live to very old lives. It's not necessarily what they eat. It's not racial, or it's not necessarily gender even. But to me, what unifies all of them, and that's an interpretation from several groups, is their social structure. That especially when they're old, they're not alone, but they have a community, and I think many of them are probably active in their own way. They still do something and are participating, and they're not just sitting in a nursing home and have nobody, and they just stare at a TV the whole day. So I think there is truth in that, but it's of course hard to prescribe that to a person and then hope that this will actually reverse or treat the disease. But I think it's a process that accumulates over, decades, that type of lifestyle.
Question right here.
It seems as though there is a time dependence that's lurking in one of these basic levels. That one of these very effective factors somehow interacts with its chemical environment and exhibits some time dependence of statistical mechanics, origin, or whatever. But has that been explored?
I'm not sure I understood your question.
I'm assuming that there is a time dependence that's lurking in one of these very basic attributes, one or more, several of these factors, which is essentially the aging of that particular factor in its context, in its neighborhood. I guess I could ask, the concentration is not what you are finding to be the driver of any benign effect.
Yeah, I think a lot of these factors change in their concentration, and they're of course all constantly replenished. So each factor lives only for a short period of time, but is constantly reproduced in the body. Proteins live anywhere from a few minutes to hours, or some live days or even weeks, but most of them have relatively short half-lives. So the levels that you get for a given factor in the blood is the accumulation of how much you make and how much degrades. So if you get more with age, you either make more or you don't clear it as quickly, and that's something for each factor one has to figure out.
Right here.
Thank you. I just have one question about the experiment with the mice. The older mice, I looked at the chart, and if I can visualize it, it showed that after the third day, even the controls started to learn. And then the fourth day, it even went further, even with the Z-score or whatever, was showing that it was still going down. Had you tried like 5 days, 6 days, and what would happen? Sorry to get too into the methods, but I was just curious. Thank you.
Yeah, that's a good question. So I showed you one type of test here, and you can actually tailor this test. Like if you go to a memory clinic and they give you a test, they can tailor the test based on your skills. Some are standardized tests where you don't do that, but you often do it. So we do the same thing with the mice. First we figure out how smart are they already or naturally, and there's differences from strain to strain and how old they are. So we want to make the test difficult enough that they don't all master it, but we want to make it easy enough that some of them can achieve it, right? So you have to find this balance that you actually get the biggest difference between treated and untreated. But absolutely, you're correct. If you give them the same test again and again, they learn and they get smarter and smarter. Some will just, if there is no capacity to form a memory, it just doesn't work. And again, it's in patients with Alzheimer's disease also. If you do the testing repeatedly at the earlier stages, the patients get actually better at it. There's even some, I heard, especially high-performing individuals, they practice the test before they go to see the doctor, because the tests are online. You can figure them out. Their MMSE test or the MOCA test. And so some people practice it. That, of course, defeats the purpose, but yes, you can test them. But then once you're really demented, there's just nothing there.
There's one question here.
You said that you can determine biological age by blood sample. So I was wondering if that can be part of annual physical for every person so that we can be more proactive. We can determine from one year to another how accelerated the aging has been or not, and then address it accordingly.
Absolutely, yeah. I think that would be the future sort of personalized medicine, right? But you only want to do that if you actually know what to do about it. And if that, so we really need to prove, first of all, that we tell you it's the biological age, right? Because we don't want to be wrong. We don't want to tell you, oh, you look like a 20-year-old. You don't have to worry. And 2 years later, you have some severe disease. So we have to really test whether these factors predict correct what we want to do. And then we have to understand what we should do about it. What are the interventions? Are there interventions like you mentioned, where you exercise it, you socialize more, or whatever interventions that you can do? Now we can all do these already, but would that push us a little stronger, because the doctor would, like the cholesterol, would tell you, oh, you have to exercise a little more, yeah, now it's good. But you could potentially also develop a treatment based on these and use that as a readout for the treatment. Absolutely.